Alrighty, we're going to do section 10.2, and this will be part 1 of section 10.2. In the last section, we talked about cell division happening whenever a cell gets too big, and so cells continue to divide even after you're finished growing, and whenever you are injured, you need more cells. It triggers your cells to grow and divide and make more cells. So here the key question is, what role do chromosomes play in cell division? And chromosomes make it possible to separate our DNA precisely during cell division. And as you can see here in this picture, the chromosome is actually a very coiled up piece of your DNA. So this is our DNA right here, and the DNA gets coiled up into a chromosome, like a familiar shape here that we have of a chromosome. These chromosomes carry genetic information that is passed from one generation to the next. Every cell must copy its genetic information before cell division begins. So we have to make a copy of the chromosomes before we can divide the cells and make new cells. Each daughter cell, now a daughter cell is the resulting copy of the result of cell division, and each daughter cell has a copy of the genetic information, an exact copy of the genetic information that the parent cell, the original cell, has. And in every organism, they have specific numbers of chromosomes that differ from one organism to the next. This chart is showing us that organisms have different numbers of chromosomes. Humans have 46 chromosomes, and some organisms, like a field horsetail, which is a plant, have 216 chromosomes. So we have a wide range of numbers of chromosomes of different kinds of organisms. Now remember that prokaryotes are cells that are very simple. They are cells like bacteria, and they don't have a nucleus. Their DNA is found in the cytoplasm. And for most prokaryotes, their DNA is a single circular molecule or a single chromosome that has all the information in one chromosome. So they just have one chromosome in bacteria, like prokaryotes. In eukaryotic cells, chromosomes are located, we know, inside the nucleus, and the chromosome is made up of something we call chromatin. So chromatin is composed of two parts. It's composed of your DNA, and it's composed of these special proteins called histones. The DNA coils up around these histones to help keep it from getting tangled. So it coils around the histones to form something called a nucleosome. These nucleosomes coil some more, like this, and then those coils make more coils, and those coils coil some more, and they make what we call supercoils, and that coils up some more and makes the actual chromosome. So the chromosome is a coil upon a coil upon a coil, and it coils in a very regular pattern, a very unique pattern to keep the DNA from getting tangled up. All right, the cell cycle is made up of three parts. There is the first part where the cell grows, the second part where we are preparing for division, and the third part where we're actually dividing to form two new cells. So there's three parts to cell cycle. In prokaryotic cells, the cell cycle is a regular pattern of growth first, then DNA replication, and then cell division. Once cells have grown to a certain size, then they get a trigger or a signal to begin to replicate or copy their DNA. Then once DNA replication is complete, they divide through a process called binary fission. So binary fission is simply the process of cell division. This cell division or binary fission in prokaryotes is actually a form of reproduction. This is asexual reproduction, and we get two cells that are genetically identical. So we start out with one cell, and that one cell then duplicates its DNA. And once the DNA is duplicated, the cell divides, and we end up with two new cells, and those two new cells are exactly the exact copy of that original cell. Now, in the eukaryotic cells, those are the kinds of cells that we have, there's actually four parts. There's G1, S, G2, and M. So these are the different parts of the cell cycle. Interphase is known as the time between cell divisions, and it's made up of the G1, S, and G2 phases, and that we can see right here. This part is the interphase portion of the cell cycle. And then the M phase is the actual part where we actually divide the cell, and that's called the cell division phase. Now we want to talk about what happens during each of the parts of the interphase, the different cell parts. The first is the G1 phase. The G1 phase is known as the growth phase, so G for growth, and the cells are increasing in size, they're making new proteins, they're making organelles, and we're just growing and being a cell and doing our normal cell jobs. During the S phase, S stands for synthesis, and during the synthesis phase we make a DNA. So we're making a copy of our DNA, we're synthesizing or making a copy of the DNA, and we're replicating the chromosomes. So making a copy, so we have two copies of the DNA inside the cell by the end of the S phase. During the G2 phase is another growth phase. During that time period, we're making organelles and molecules that we need for the actual process of cell division. So it's our last stage before we actually divide the cell, and it is preparing for the cell division. So we're another growth phase, but we're in that prep phase. The M phase is known as the mitosis phase or the cell division phase, and it's made up of two parts. There is first the mitosis phase, which is actually dividing the nucleus, and then there's cytokinesis, which is the dividing of the cytoplasm. And in part two, we'll be focusing primarily on the M phase of the cell division phase, where we have the parts of mitosis and also then cytokinesis.